Welcome to the deep dive. Today, we're plunging into a topic that, well, while undeniably tragic in its human impact, offers some of the most profound insights into fundamental cell biology: cancer.
Right. Sort of a paradox, isn't it? Studying how things go wrong actually helps us understand how they're supposed to work, the normal rules, how they're enforced.
Exactly, and how they break down in cancer. Our mission for this deep dive is to distill the most vital insights from a really comprehensive chapter on cancer in molecular biology of the cell.
Yeah, it covers a huge amount of ground from, the basic nature of the disease right through to prevention and the latest treatments.
And we promise to try and simplify these complex molecular and cellular processes, make them understandable, make them relevant to you without skipping the crucial details. Okay, let's unpack this. Imagine your body as this incredibly organized city, right? A bustling society of trillions of cells, each doing its job.
A society, yeah. But with this peculiar rule, self-sacrifice. It's not survival of the fittest cell. It's cooperation for the good of the whole organism.
Which is counterintuitive in a way.
Yeah.
So how does cancer hijack that fundamental principle? How does it turn harmony into, well, chaos?
It really comes down to how committed those cells are to collaboration.
Hmm.
Their behavior, it's all tightly regulated by signals from outside the cell, these sort of social controls.
Telling them when to grow, when to rest, when to die.
Exactly. All for the good of the organism. But, mutations happen constantly. Billions every day in a human body.
Hmm.
Most are harmless.
But occasionally.
Occasionally, a mutation disrupts that harmony. It gives one cell a slight edge, maybe grows a bit faster or survives when it shouldn't. And that cell can become the founder of a rebel clone.
Mm-hmm.
And through more mutations, competition, natural selection, it's like evolution and fast forward happening inside you. This clone evolves, becoming more aggressive, and eventually threatens the whole organism. That's really cancer at its core, a microevolutionary process.
Wow, so it's literally evolution happening inside our bodies. Okay, so if it's evolving, what defines these rebel cells? What makes them a true cancer, not just some weird misbehaving cell?
That's a really critical distinction. Cancer cells have two defining heritable properties.
Mm-hmm.
First, they reproduce without the normal restraints. They just keep dividing.
Ignoring the stop signs.
Right. And second, crucially, they invade and colonize territories that belong to other cells. That's second ability, that's metastasis.
And that's the really dangerous part.
That's usually what's lethal. See, an abnormal cell that just proliferates uncontrollably forms a tumor or neoplasm. If those cells haven't invaded yet, it's benign, often easily cured.
Okay.
But it's only considered a true cancer if it's malignant. That means its cells can invade surrounding tissue and spread, forming secondary tumors, metastases. Looking at figure 20.1 really illustrates that spread. That's what typically kills the patient.
So looking at the bigger picture then, where do most cancers actually start, and why do we see certain types, like, way more often?
Well, they're classified by the tissue and cell type they come from. And about 85%, the vast majority, are carcinomas.
Carcinomas. Right.
These arise from epithelial cells, the cells lining our organs, skin, gut, lungs.
Why so many there?
Makes sense, actually. Epithelial tissues have high cell turnover, lots of division, so more chances for mutations. Plus, they're often exposed to damage from the environment, and think UV radiation on skin or chemicals in the lungs or gut.
Makes sense. What about the others?
Then you have leukemias, lymphomas, myelomas, cancers of blood cells, and sarcomas, which are less common, from connective tissue or muscle. Figure 22 shows the incidence and mortality rates. It's quite striking.
And the naming, like adenoma versus adenocarcinoma.
Right, that reflects benign versus malignant. An adenoma is a benign gland-like tumor, like a colon polyp, which you can see in figure 23. An adenocarcinoma is its malignant invasive version.
Okay, here's something I've always found hard to grasp. How can we be so sure that a massive tumor, maybe years old with billions of cells, started from just one abnormal cell? What's the actual proof?
It's really compelling evidence, actually. Most cancers are indeed thought to have a clonal origin. One cell gets that first hit, that heritable change. Then its descendants accumulate more changes.
But how do we prove it came from just one?
Molecular forensics, essentially. Take chronic myelogenous leukemia, CML. Almost all patients have leukemic cells with a specific defect. The Philadelphia chromosome.
The chromosome swap.
Exactly, a translocation between chromosome 9 and 22. If you sequence the DNA right at that breakpoint, that specific join, it's identical in every single leukemic cell in that person.
Wow.
But it's different between different patients, like a unique fingerprint for that initial event. Figure 25 shows this beautifully. It proves they all came from one ancestral cell where that specific accident happened.
And these are mutations in body cells, right? Not inherited.
Mostly somatic mutations, yes. Not usually inherited. The inherited mutations can predispose you. And we now know epigenetic changes altering gene activity without changing the DNA sequence are also huge drivers. It all links back to heritable changes in the cell.
Okay, but if billions of cells mutate daily, why isn't cancer, like, everywhere, all the time? It feels like one mutation should be enough to kick it off much more often.
That's a great question. And thankfully, one mutation is almost never enough. If it were, cancer would likely be a childhood disease for everyone.
Right.
The evidence points to needing a substantial number of independent, rare genetic and epigenetic accidents to accumulate in one cell lineage.
So multiple hits are needed.
Exactly. Look at epidemiological data. Figure 20:6 shows how cancer incidence rockets up with age. That wouldn't happen if one hit was sufficient.
It suggests accumulation over time.
Precisely. And think about lung cancer and smoking. There's a long lag time, often decades, between starting smoking and getting cancer, as shown in Figure 20:7. It takes time for that damage, those mutations, to build up. It's a gradual process called tumor progression, like Darwinian evolution in miniature. Figures 20:8 and 20:9 illustrate this stepwise progression.
But evolution isn't always slow, is it? Sometimes there are these sudden massive changes. Does that happen in cancer too, like a genomic Big Bang?
It absolutely does. While gradual accumulation is common, cancer evolution can also have these abrupt, catastrophic leaps.
Right.
Some call it the Big Bang model.
What drives that?
Well, most human cancer cells are genetically unstable. They accumulate changes much faster than normal cells. This instability actually helps them evolve faster.
So chaos speeds things up.
In a way, yes. If you look at the karyotype, the chromosome set, of many cancer cells, it's often a total mess. You see breaks, rearrangements, deletions, duplications. Figure 2010 shows some examples. Often they have aneuploidy, the wrong number of chromosomes, from errors in division.
And the Big Bang part.
That can come from something even more dramatic, like chromothripsis. Imagine a chromosome gets isolated in a little side nucleus, a micronucleus. It gets shattered into pieces, then randomly stitched back together. Figure 2011 tries to depict this chaos.
Like smashing a vase and gluing it back randomly.
Exactly. It creates completely new, potentially advantageous gene combinations in one catastrophic event, accelerating progression.
Okay, and within this evolving, sometimes chaotic population, there's this idea of the cancer stem cell. What makes them special, and why are they such a headache for treatment?
Right. This is really important. Think about tissues that constantly renew themselves. Gut lining, skin, bone marrow. These rely on stem cells.
The ones that can make more of themselves and other cell types.
Exactly. They self-renew and produce transit-amplifying cells that divide rapidly for a while, then differentiate. Figure 2012 shows that tissues with higher division rates tend to get more cancer.
Okay.
Now, in some tumors, there seems to be a similar hierarchy, but it's skewed. There's a small population of cancer stem cells shown conceptually in Figure 2013. They can self-renew indefinitely and sustain the tumor's growth.
And the problem is?
The problem is these cancer stem cells are often relatively slow dividing compared to the bulk of the tumor. Traditional chemo and radiation target rapidly dividing cells.
So they kill the bulk, but miss the root.
Precisely. The treatment might shrink the tumor dramatically, but if it leaves behind those resistant cancer stem cells, they can just regrow the tumor later. It's a major reason for relapse.
Okay, so we've got this picture of evolving, unstable cells. What does this actually mean for how they behave? The chapter talks about these acquired superpowers or hallmarks of cancer cells. Let's walk through those key characteristics.
Yeah, they acquire a set of, you could call them subversive new skills that let them thrive and spread.
So what's first?
First, altered homeostasis. Basically, more cells are produced than die off. This could be faster division or, very often, less apoptosis programmed cell death. Figure 2014 shows this balance shift. They just refuse to die when they should.
Okay, what else?
Second, they bypass normal proliferation limits. Normal cells show contact inhibition, right? They stop dividing when they bump into each other. Cancer cells, like you see in Figure 2015, lose that, they pile up.
And they keep dividing indefinitely.
Many do. They overcome replicative senescence, the normal limit on cell divisions. Often by reactivating telomerase to maintain the ends of their chromosomes, the telomeres, they become effectively immortal.
Resisting death signals seems key too.
Absolutely. That's the third hallmark, evasion of cell death signals. Our cells have built-in suicide programs for when things go wrong. Cancer cells have to disable those safety nets.
Though you still see cell death in tumors.
You do, especially in the core of large tumors where conditions are harsh, lack of oxygen, nutrients. But that's often necrosis, messy cell bursting, as shown in Figure 2017, which can actually promote inflammation and tumor growth, unlike clean apoptosis.
And they change how they eat, right? The Warburg effect.
Yes. Fourth, altered cellular metabolism. It's fascinating. They guzzle glucose way more than normal cells. But even with plenty of oxygen, they prefer a less efficient energy pathway. This is the Warburg effect illustrated in Figure 2018.
Why do that? Seems inefficient.
It seems counterintuitive for energy, but it's great for building blocks, fats, proteins, DNA precursors. They prioritize rapid growth and division over just making ATP efficiently.
Clever. And they manipulate their surroundings.
That's fifth. Manipulation of the tissue environment. A tumor isn't just cancer cells, it's interacting with the tumor, stroma, fibroblasts, immune cells, blood vessels, like we see in Figure 2019.
How do they manipulate it?
They induce angiogenesis, tricking the body into growing new blood vessels to feed the tumor. And critically, they create an immunosuppressive microenvironment, basically hiding from or actively blocking the immune system.
And finally, the deadliest hallmark.
Metastasis.
Right.
Responsible for about 90% of cancer deaths. It's a whole cascade, shown schematically in Figure 2020. Invade local tissue, get into circulation, travel, get out somewhere else, extrovasation. And then the hardest step, colonize that new site. Very few cells succeed in colonization.
Is that where EMT comes in? Epithelial to mesenchymal transition?
Potentially, yes. EMT might give epithelial cancer cells the ability to detach, become migratory, and invade, which is crucial for metastasis to begin.
Okay, so we know what they do. Now, how do they gain these abilities? This brings us to the cancer-critical genes.
Right. These are the genes whose alteration drives the cancer process. They broadly fall into two main classes. Think accelerator and brakes.
Accelerator first.
Those are the proto-oncogenes, normal genes involved in growth signaling. When they get a gain-of-function mutation, they become hyperactive oncogenes, like a stuck accelerator pedal. Usually, mutating just one copy is enough. It's dominant. Figure 2021a shows this.
And the brakes.
Tumor suppressor genes. These normally restrain growth. You need a loss-of-function mutation in both copies to contribute to cancer, like losing both sets of brakes. It's recessive at the cellular level. Figure 2021b illustrates this. There's also a subgroup that maintains the genome DNA repair genes. Losing them ramps up the overall mutation rate.
The discovery of these genes must have been quite a journey. Any key stories?
Absolutely. Oncogenes were first really understood through animal retroviruses, like the rhesusarcoma virus carrying VSRC. That was a mutated version of a normal cellular gene, CSRC.
But most human cancers aren't caused by viruses like that.
Exactly. The huge breakthrough was in the early 80s. Researchers found that transferring DNA from human bladder cancer cells into normal mouse cells could transform them, the culprit. A mutated human Ray's gene.
The Ray's oncogene.
Yes, and it wasn't a virus, just a single-point mutation making the Ray's protein hyperactive. That showed human cancers arise from mutations in these same critical genes. RAS mutations are now found in maybe 30% of human cancers.
How else can proto-oncogenes become oncogenes?
Several ways, shown in Figure 2022. Point mutations, like with Ray's or the EGF receptor in glioblastoma, shown in Figure 2023. Gene amplification, making many extra copies of the gene, or chromosomal rearrangements, creating fusion proteins or putting the gene under control of a strong promoter, like MYCEC in Burkitt's lymphoma.
And tumor suppressors, how were they found?
Often through studying rare hereditary cancers. Retinoblastoma, the eye cancer, was key. Kids with the hereditary form inherit one bad copy of the RB gene. They only get cancer if the second good copy is lost in a retinal cell the second hit. Figure 2024 explains this beautifully.
The two-hit hypothesis.
Exactly. Non-hereditary cases need two separate somatic hits in the same cell. The RB protein itself is a crucial break in the cell cycle, and its loss is common in many adult cancers too.
How do these tumor suppressors get knocked out?
Various ways, shown in Figure 2025. Small deletions, point mutations, losing a whole chromosome, but also, importantly, epigenetic silencing. The gene is still there, sequence unchanged, but it's switched off, often by DNA methylation. This is a really common way to lose tumor suppressor function.
It's amazing how technology, especially sequencing, has just blown open our view of the cancer genome.
Completely transformed it. We went from finding individual genes to sequencing the whole genome, or exome, the protein coding parts of tumors. We can find point mutations, copy number variations, rearrangements, everything.
Comparing tumor to normal tissue from the same person.
Exactly. Plus, RNA sequencing tells us what's expressed. Mass spectrometry looks at proteins. Huge projects like the Cancer Genome Atlas, TCGA, and COSMIC have cataloged alterations across thousands of tumors. It gives us an incredible landscape view. We can even see distinct mutation patterns in oncogenes versus tumor suppressors, as Figure 2026 highlights.
And what does this landscape look like? Is it just a few key mutations or more like total chaos?
Often it's pretty chaotic. Many cancers have wildly disrupted genomes. Some have relatively normal chromosome sets, but tons of point mutations, usually due to faulty DNA repair. Others show massive aneuploidy and rearrangements like we see in Figure 2027, suggesting those catastrophic events like chromothripsis.
And we can sometimes pinpoint the underlying cause.
Yes. Defects in DNA repair genes like BRCA1 and BRCA2 are linked to specific patterns of breaks, especially in ovarian cancer. And it's not just DNA sequence. Epigenetic changes are crucial. Maybe half of cancers have mutations in chromatin-modifying proteins. Figure 2028 emphasizes this interplay. Sometimes a genetic mutation, like in the IDH gene, directly causes widespread epigenetic changes, like DNA hypermethylation.
But amidst all this complexity, hundreds of mutated genes, you're saying many cancers still rely on messing up just a few core pathways.
That's one of the most profound insights, yes.
Yeah.
We need to distinguish the driver mutations, the ones really pushing the cancer forward, from the passenger mutations that are just along for the ride. And the drivers, though numerous, tend to converge on disrupting just 3 fundamental control pathways. Figure 2029 lays these out.
Okay, what are the big 3?
First, the RB pathway, controlling cell cycle entry. Got to break that break. Second, the RT-CRASPI3 kinase pathway. This governs cell growth and proliferation. It's almost always hyperactive in cancer, driving that Warburg metabolism too, as shown in Figure 2030. Losing the PTN suppressor, which opposes this pathway, is very common.
And the third?
The P53 pathway. Absolutely critical. P53 is like the cell's emergency response system, shown in Figure 2031. It senses stress DNA damage, low oxygen, oncogene activation, and triggers cell cycle arrest, senescence, or apoptosis. It's the guardian of the genome.
So you need to disable the guardian too.
Pretty much always. Almost every cancer has mutations disabling each of these 3 pathways, RB, RASBR3K, and P53. And interestingly, usually just one key mutation per pathway is enough. It's about disrupting the pathway's function, not necessarily hitting every gene in it.
How do researchers confirm these complex interactions work this way in a living organism, not just in a dish?
Mouse models are incredibly powerful for this. You can engineer mice to express oncogenes or lack tumor suppressors, often in specific tissues or switched on at will, as shown in Figure 2032.
What are they shown?
They confirm that single changes usually aren't enough. For example, mice with just an activated myfoot or res oncogene get cancer, but slowly. Mice with both, as Figure 2033 shows, get aggressive cancers much faster. It really proves the need for multiple hits.
And they help study tumor diversity.
Yes, sequencing different parts of a tumor or its metastases reveals that tumor heterogeneity. It's Darwinian evolution happening within the tumor, generating subclones. And now, using human organoids mini-tumors grown in the lab from patient cells, researchers can even reconstruct the evolutionary tree of mutations, like they did for colorectal cancer in Figure 2034. It's amazing detail.
Let's use colorectal cancer as a specific example then. It seems like a classic case of step-by-step progression.
It really is a great model system. Progression from benign adenomatous polyps, like in Figure 2035, to malignant carcinoma is well studied and can take years. Colonoscopies can catch and remove those precursor polyps.
What are the key genes involved there?
The big 3 are ATPAC, mutated in over 80% of cases, often very early. Then KRAS, activated in about 40%. And P53, inactivated in about 60%, usually later.
What does APAC do?
Its role was nailed down studying FAP, the hereditary polyp syndrome. People inherit one bad APAC copy. APC protein normally helps degrade PADNA. Loss of APC means too much apatinin, driving proliferation, especially of gut stem cells. Table 20:1 lists key genes. There's also HNPCC, another hereditary form caused by defects in DNA mismatch repair genes like MLH1. Those tumors have tons of point mutations.
So is there a typical order for these mutations in colorectal cancer?
A common sequence, shown in Figure 2036, seems to be APAC inactivation first, leading to early polyps, then KRAS activation, often seen in larger polyps, and P53 inactivation usually comes later, allowing cells to survive the stress of hyperactive oncogenes and become truly invasive.
But it's not always that exact order.
No, the sequence can vary, and how metastasis really happens, especially at the colonization step, is still a bit of a black box. Epigenetic changes, maybe EMT, likely play big roles there too.
Okay, so this incredibly detailed understanding isn't just academic. It must be changing how we approach cancer prevention and treatment. Let's start with prevention feels like the ultimate goal.
Absolutely. And the encouraging thing is a huge chunk of cancers are preventable. Look at studies of migrant populations shown in Figure 2037A. When people move, their cancer rates tend to shift towards those of their new home country. That points strongly to environmental factors.
So lifestyle and environment, not just genes.
Definitely. It's estimated maybe 45% or more of cancer deaths are linked to modifiable risk factors, things we can potentially change. Figure 2037b breaks some of these down. Over half might be avoidable.
How do we even identify what in the environment is risky and what are the biggest culprits?
We use tests like the Ames test to screen chemicals for mutagenicity, their ability to damage DNA. But many chemical carcinogens aren't directly harmful. They have to be activated by our own liver enzymes, cytochrome P450s.
Like what?
Things like benzoamperine from burning organic matter or aflatoxin from moldy food, shown in Figure 2038, they get converted into DNA-damaging forms. But the single biggest culprit globally, tobacco smoke, no question.
And beyond smoking.
Other major factors, illustrated in Figure 2039, include excess body weight, alcohol, poor diet, lack of physical activity. These lifestyle factors contribute significantly.
What about infections? Can things like viruses actually cause cancer?
Yes, definitely. Infectious agents, viruses, bacteria, parasites cause about 15% of cancers worldwide, even more in developing regions. Table 22 lists some key ones.
How does that work?
Some DNA tumor viruses, like HPV, human papillomavirus, directly cause cancer. HPV makes viral proteins, E6 and E7, that specifically target and inactivate our own P53 and RB proteins. Figure 2040 shows this mechanism. That's why HPV vaccination is so effective at preventing cervical cancer.
So they disable our defenses. Any other ways?
Sometimes it's indirect. Chronic inflammation from hepatitis B or C viruses can lead to liver cancer. The bacterium Helicobacter pylori causes inflammation leading to stomach cancer. Even HIV contributes indirectly by weakening the immune system, allowing other cancer-causing viruses like the one behind Kaposi sarcoma to thrive.
Okay, shifting to treatment. The search for cures has been long and tough. Why is cancer so hard to actually get rid of?
It's incredibly challenging. You have to eliminate every single cell, including hidden metastases. Traditional treatments are often toxic to normal cells too, causing side effects.
Yeah.
And the biggest problem, cancer involves resistance. Its genetic instability allows it to adapt and survive treatments.
So how do the traditional therapies work, and how are the newer, more targeted approaches different?
Well, traditional radiotherapy and cytotoxic drugs mostly work by causing DNA damage or messing up cell division. They tend to kill cancer cells more effectively because those cells often have faulty checkpoint controls and DNA repair, so they blunder into cell death when damaged.
Yeah, but they hit normal cells too.
They do, especially rapidly dividing ones like in the gut or bone marrow, causing side effects. And resistance is a huge issue, often linked to losing P53 function, which prevents damaged cells from undergoing apoptosis.
Which leads to the need for targeted therapies.
Exactly. The ideal is synthetic lethality. Find a drug that only kills cells with a specific cancer-causing defect but spares normal cells.
Like the PRP inhibitors.
PRP inhibitors are a prime example, illustrated beautifully in Figure 2041. Cancers with faulty Brexaco or Brexaho genes can't properly repair DNA double-strand breaks using homologous recombination. PRP inhibitors block a different repair pathway for single-strand breaks.
So you knock out two pathways in cancer cells, but normal cells still have a backup.
Precisely. The combination is lethal only to the BRCA-deficient cancer cells. Normal cells shrug it off. It's shown amazing results in certain breast and ovarian cancers with fewer side effects.
What about targeting the oncogenes themselves?
That's another major strategy, designing small molecules to block specific oncogenic proteins. The poster child here is imatinib, or Gleevec, for CML.
The drug for the Philadelphia chromosome?
Yes. CML is driven by that hyperactive BCR-ABL fusion protein, Figure 2042. Imatinib was designed to fit perfectly into BCR-ABL and shut it down, as shown in Figure 2043. The results were revolutionary for CML patients.
Does resistance still happen?
It can, through secondary mutations in BCR-ABL. But then researchers developed second-generation inhibitors to overcome that. It shows the power of targeting the specific driver. We now have other kinase inhibitors against targets like EGFR or BRAF for other cancers, seen in Figure 2044. It's a whole new era.
And beyond drugs hitting cancer cells directly, what about using our own immune system?
Immunotherapy. It's incredibly exciting. One approach uses antibodies, like trastuzumab, Herceptin, targeting HER2-positive breast cancers. Or antibodies can be used to deliver toxins specifically to cancer cells.
But getting our T cells involved seems even more powerful.
It is. Our cytotoxic T cells can recognize abnormal proteins, neoantigens, on cancer cells that arise from mutations, as shown in Figure 2045. Adoptive cell transfer expands a patient's own tumor-fighting T cells in the lab and reinfuses them.
And CAR-T therapy.
CAR-T therapy engineers a patient's T cells with a synthetic receptor, a CAR, to make them recognize and kill cancer cells much more effectively. It's been a game-changer for some leukemias and lymphomas.
But tumors fight back against the immune system, right?
They do. They create that immunosuppressive environment. A key trick is expressing proteins that engage immune checkpoints on T cells, like PD-L1 binding to PD-1. This acts like a brake, shutting down the T cell attack, as shown in Figure 2046A.
So checkpoint inhibitors release the brakes.
Exactly. Drugs that block PD-1 or PD-L1 take the foot off the brake, unleashing the T cells. These have shown remarkable success, especially in tumors with lots of mutations, because they have more neoantigens for the T cells to see. Some tumors also block T cells from even getting in, maybe by interfering with dendritic cells, like in Figure 2046B.
But even with these advances, the battle isn't over. Cancer keeps evolving resistance.
That remains the core challenge. That same genetic instability that makes cancer vulnerable also allows it to evolve resistance. BRCA cancers can mutate again to resist PIRP inhibitors. Tumors can overproduce drug pumps like MDR1. A few resistant cells survive treatment and regrow.
So what's the path forward?
Combination therapies are key, hitting the cancer from multiple angles at once. And individualized treatment, using detailed genomic analysis of each patient's tumor to choose the best combination.
So the tools we have now to understand the tumor are crucial.
Absolutely. We can sequence tumors, understand their specific vulnerabilities. Models like human tumor xenografts in mice or those organoids grown from patient cells allow us to test therapies personalized to that specific tumor.
It sounds like progress is happening, even if it feels slow sometimes.
It is. Clinical trials take time, but the systematic approach, grounded in this incredibly deep knowledge of basic cell biology, how cells divide, signals survive, die, interact, and understanding how cancer subverts all of that, it gives us real reason for optimism. We're learning how to use that knowledge more precisely every day. #outro.
And that brings us to the end of our deep dive into the molecular biology of cancer. It's just incredible, isn't it, how studying this disease reveals so much about the fundamental rules of life itself.
It really is. What's fascinating is this constant interplay, the sort of arms race between our understanding and cancer's ability to adapt. Every breakthrough, every new therapy teaches us more not just about fighting cancer, but about the incredibly intricate dance of life, death, cooperation, and rebellion inside our own cells.
Absolutely. It really makes you appreciate the complexity and, I guess, the fragility of that cellular society. A really profound thought. Thank you for joining us on this exploration today.
My pleasure.
We hope this deep dive has given you a powerful shortcut to being truly well-informed about one of biology's most complex, challenging, and ultimately revealing subjects.